स्टेप नंबर 1 के हवाले से हमने बात कंप्लीट की कि हमारे पास सोर्स ऑफ सैंपल के लिहाज से हमें स्टेप नंबर 1 अडॉप्ट करके सेल लाइजेट्स और होमोजिनेट्स प्रिपेयर करने हैं. Once these cell lysates or homogenates are prepared, we proceed further for isolation and purification of RNA with the subsequent steps. इस मॉड्यूल में स्टेप नंबर 2 से स्टार्ट लिया है बिकॉज स्टेप नंबर 1 हम ऑलरेडी डिस्कस कर चुके हैं और स्टेप नंबर 2 में हमने अब नेक्स्ट बात करनी है दैट द होमोजेनेट्स विच वी हैव ऑब्टेन्ड फ्रॉम step number 1, they should be incubated for 5 minutes at room temperature so that the nucleoprotein complex can be dissociated. इस dissociation को further ensure करने के लिए हम pressure method भी use कर सकते हैं. जैसा कि पहले मैंने जिक्र किया, एक needle and syringe use करते हुए हम lysate को needle में fill in करें और प्रेशर के साथ पुश आउट करें इन आर ट्यूब, प्लास्टिक ट्यूब. और प्रेशर इतना ज़्यादा नहीं होना चाहिए कि हमारा लाइजेट जो है वो स्प्लैश आउट करके ज़ाया हो जाए. इस किस्म की फोर्स पासिंग थ्रू द नीडल विल इंश्योर दैट द न्यूक्लियोप्रोटीन कॉम्प्लेक्स आर डिसोसिएटेड एंड देयर आर नो सच कॉम्प्लेक्सेस एंड द RNA इज नॉट रिटेन्ड बाय एनी अनवांटेड मॉलिक्यूल. इस स्टेप को टू पुश इन, टू टेक इन द लाइजेट एंड टू पुश आउट द लाइजेट कैन बी रिपीटेड फॉर 5 times. After this denaturation, now this is just time to add chloroform or the second phase. The chloroform is added as per our initial volume of the monophasic reagent. 0.2 ml of chloroform is added per milliliter of the monophasic reagent. So we have to go back to our step number 1 when we were preparing the lysate. What was the volume of monophasic lysis reagent we used? Accordingly, we calculate the volume of chloroform required. Then this mixture is vigorously mixed by using vertex mixer. Because once the chloroform is vigorously mixed up with the cell lysate, then it is going to trap or it is going to dissolve the proteins and the DNA is separated from the aqueous phase. Once this vigorous mixing is done, the next step would be to separate the two phases, the aqueous phase and the organic phase. And this separation can be done by centrifugation at 12,000 RPM for 15 minutes at 4 degree centigrade. 4 degree centigrade के ऊपर जब ये सेंट्रिफिकेशन का स्टेप किया जाएगा तो ऑर्गेनिक फेज और एक्वस फेज ऑटोमेटिकली सेपरेट हो जाएंगे. अब ये जो टॉप एक्वस फेज है दैट कंटेन्स द RNA, बॉटम ऑर्गेनिक फेज कंटेन्स द प्रोटीन और जो इनका इंटरफेस है दैट कंटेन्स द DNA. So very carefully we have to transfer the upper aqueous phase into a new tube. So we should ensure that we don't take any DNA along with the aqueous phase so as to avoid the contamination of RNA with DNA. This aqueous phase is further used for RNA precipitation, and this precipitation is done using isopropanol 0.25 volume as per the monophasic lysis reagent. If we use 1 milliliter, we are going to use 0.25 ml of the isopropanol and 0.25 volumes of RNA precipitation solution. So for precipitation, we are going to add isopropanol 0.25 volume. We are going to add RNA precipitation solution 0.25 volume of the initial monophasic lysis reagent. Mix it thoroughly, store it at room temperature for 10 minutes and then proceed to the collection step, the collection of the precipitated RNA. It is done by centrifugation, 10 minutes, 4 degree centigrade. इस centrifugation step के बाद RNA is precipitated at the bottom. Supernatant is no more required. It is discarded. And the next step comes the washing of this RNA so that any solution traces left behind should be washed away. And this washing is done by 75% ethanol. अब ethanol से हम 2 times washing करते हैं और जो ethanol है उसको remove करने के लिए we do the same step. We do the centrifugation. RNA settles at the bottom. Ethanol is no more required. It is aspirated or it is taken out by using the pipette, the micropipette. Any traces of the ethanol can be removed by using the disposable pipette tip or by letting the tube open standing at the working bench, so that because of evaporation all the ethanol gets evaporated and no ethanol traces should be left along with the RNA. The next step would be to solubilize or to dissolve this RNA into DEPC treated water. That volume of that water depends on our requirement, how concentrated RNA we want, how much tissue weight or the number of cells we took to isolate this RNA. Normally we use 50 to 100 microliter of the DEPC treated water to suspend the or to dissolve this precipitated and purified RNA. This RNA solution can be stored at -70 degrees centigrade for further usage. Before using this RNA for further processing, like for gel electrophoresis, for reverse transcription, which will be preparing the cDNA for usage in this PCR or any other process, the concentration estimation is required, and the concentration is determined using UV spectrophotometer at 260 nanometer and 280 nanometer absorbance. यहाँ पे एक बात और मैं बताता चलूँ कि RNA concentration estimation के लिए हमें RNA को dilute करना पड़ता है, keeping in view the volume of the cuvette. कितनी volume उस quartz cuvette में accommodate कर सकती है. We dilute the RNA sample 100 times or 200 times. That means if we take 1 microliter of RNA, then we add 99 microliter of water, the DEPC treated water, so that our main volume of RNA should not be wasted. Instead of using 1 microliter, we use 100 microliter to concentration determination, then nothing is left behind for further processing. So to estimate the concentration of RNA, we need to dilute it 100 times or 200 times, whatever is the availability or whatever is the requirement of the researcher.